So in terms of kind of the previous material that's been covered or the previous material, the questions that have been asked, I would say of the 80 questions, I'll probably reuse about half of the questions that I've used on previous exams. So about 40 of those questions will be questions from previous exams. I'm going to open up those previous exams for you to view, review. And so that's a place I would spend probably a good chunk of my time if you feel comfortable with those questions on those previous exams. Now, because I'm allowing people to go back and correct their answers, you won't necessarily have the answers for there. So your test grades from what? From your third exam? Somebody asked, when will our test grades be up? Are you talking about for the third exam or for the final? Exam 3. Oh, okay. I guess I wasn't really aware that you weren't able to view those yet, so I can easily just change the settings in Moodle as soon as we wrap up here, so you can view the results from exam 3. Outside of getting your pro section grades entered today, the other thing I'd like to do is get all of your exams opened up so you can make those corrections, as well as viewing what you did and did not get correct on exam 3. So that's easy enough. Does anybody have any other questions about execution? How do we submit corrections? So it's basically you should be able to essentially reopen the exam that you've previously taken and then be able to make changes to the questions that you have been marked incorrect. So it'll just go to the previous exam kind of link, click on it, and then you should be able to go through and make those corrections. If for some reason that doesn't necessarily work for technical reasons, we can go back and have you submit them like via a Word document. I would like to avoid that as much as possible, but we'll see if technology is with us right now. I feel like I'm fighting an uphill battle with it, but you know, that's the nature of the times that we live in right now. I guess other announcements while folks are thinking of what questions. There were a lot of eye muscles in the study guide, but those weren't on exam 2. Will they be on the final? I wouldn't say that I'd spend a significant amount of time on eye muscles per se. Eye muscles were part of that study guide just because, you know, we were learning about those were part of the muscles that you were responsible for knowing, but not necessarily something that I would spend a ton of time with. Like I said, I'll probably go around half of previous asked questions from the exams. And then the questions that I asked that are new questions are not going to be super, I would say specific. They might even be pulling from different concepts from different chapters, pulling them together, you know, because we've talked about things like, you know, we're talked about the central nervous system and how it's sending action potentials to muscles. And so we talk about how there are specific, let's say, cranial nerves that innervate muscles that maybe we've talked about. So, you know, kind of bringing those concepts together in the big picture would probably be the types of questions that you'd see outside of those 40 questions that will be pulled from previous exams. Yeah, so it's not, my goal is not to ask you like a single point from a single sentence from a single chapter. It's much more kind of broad, big picture types of questions than you'd really be seeing elsewhere. You probably see some application questions like, you know, many of you want to be physicians or physical therapists. So I might present a patient to you and say, this patient is presenting with X, Y, and Z. What do you think that it could be given your knowledge from the course? And so hopefully by asking you questions like that, you start to see kind of why you're learning these things in this particular class. Come on, somebody has to have like specific questions about material. Chapter 2 questions. 4 or 10 or 11. So when you say chapter 2, what are you pulling from?
I'm just looking at the final exam study guide under the header chapter 2.
Study guide. Too many files open here. Okay, chapter 2, and you said questions 4, 10, or 11. So, okay, Great. Okay, so when we're talking about different types of bonds, the first question there is distinguish between among ionic, covalent, and hydrogen bonds. And so often when we think about bonding, you need to be thinking about electrons. And so electrons are going to be those negatively charged, smaller, you can't even use the word molecule, the negatively charged electrons and the electron clouds surrounding a particular type of element. And so with an ionic bond, you're going to have the complete transfer of electron from one to another, from one atom to another. And so sodium chloride is going to be probably the best example of an ionic bond, where you have sodium that has an electron that it wants to accept, and chloride that has an electron that it wants to lose. Chloride has an electron that it wants to gain, and sodium has an electron that it wants to lose. And so what's going to happen there is that you're going to have a more that movement of that electron from the sodium to the chloride is going to cause the sodium to become positively charged, that chloride to become negatively charged because of that movement of that electron, and that they're going to form an ionic bond due to their charge differences. Now, that's an example of an ionic bond. A covalent bond is going to be the sharing of electrons. And so if you have, let's say, carbon, for example, would be a great atom to use as an example, and then carbon and let's say hydrogen. So methane would be a great example of that, which is CH4, because that carbon has 4 electrons in its outermost shell, which means it has basically 4 slots that could be filled, or 4 places where it could be sharing electrons with other atoms. And so in the case of methane, CH4, you've got 4 hydrogens that are being connected to the outside there, where you have a hydrogen that's donating, or not donating, but sharing an electron, and then carbon, which is sharing an electron. And so because of that, both hydrogen and carbon get kind of happy or satisfied because they've got the right number of electrons in their outermost shell. For hydrogen, it's 2 because it only has that first shell that needs to be filled. And then for that carbon, it has, 4 bonds being made with hydrogens, which means that you have 8 electrons in that outermost shell, and that makes carbon really happy. And so the sharing of electrons between those 2 different types of atoms makes those bonds very strong. And so when we talk about bond strength, covalent bonds are going to be the strongest types of bonds, probably followed by, in the list that we just gave there, ionic bonds will be second. And lastly would be hydrogen bonds. Now, hydrogen bonds are important. They're weak bonds. They're just, hydrogen generally has, because of its electron structure, will bond with things like, let's say, oxygen, for example, and share those electrons, form a covalent bond with oxygen, and thus make things like a water molecule. And so because of that, oxygen is going to pull the electrons closer to it, and the oxygen is going to be slightly more negative, and that hydrogen is going to be slightly more positive. And then that slightly more positive hydrogen can form a hydrogen bond with a slightly more negative oxygen. And so because of that, hydrogen bonds are very weak bonds. We see hydrogen bonding also in the strands that connect the strands of DNA. Weak bonds aren't necessarily bad bonds. I think when people hear the word weak, they're like, oh, it's a weak bond, it doesn't matter. But actually, a lot of hydrogen bonds put together are very important. Like I said, they hold together the 2 strands of DNA. And it's important for those strands of DNA, kind of going back to some earlier chapters, it's important for those strands of DNA to be able to be held together very tightly, but then also in some instances be pulled apart. So when we talk about things like transcription or replication, where you have to have access to the inside of a double-stranded piece of DNA, it's important that you can break those hydrogen bonds and gain access to that material that's between that. And so, so again, kind of the order of those bonds would be, in terms of strength, would be covalent, ionic, and hydrogen. And I kind of gave some examples of each one of those. And I know I'm talking with my hands. I probably should just pull up a PowerPoint slide and start drawing, but to a certain extent, my handwriting's terrible, so I don't know how much more helpful that would be. Okay, I want to keep moving forward to make sure I get through all these questions. So question 10, identify the building blocks in the function of carbohydrates. Okay, so and then 11 it would be lipids. So for carbohydrates, it's basically talking about kind of the monomer and polymer components, and the same thing with lipids when you're talking about a monomer and a polymer. When I say monomer and polymer, I'm talking about kind of single components and then kind of a whole chain of components. And so for the case of carbohydrates, carbohydrates are ringed sugars. When you hear carb, you should just automatically think sugar. Their carbon ring structure, when we talk about carbon being so important. And so an example of just kind of like a single ring structure would be something like glucose. There's just a single carbon ring there. And then kind of a chain, generally when we're talking about those, we're talking about disaccharides. And so disaccharides would have basically two different sugars that were connected. And so sucrose is an example of a disaccharide, and glucose is an example of a monosaccharide, meaning mono, meaning one, and then disaccharide would be two. And so that's kind of an example of both just a single component and then kind of a chain of components. The functions of carbohydrates, big picture, when we're talking about the body, your body right now, like you probably just maybe had some lunch, and that lunch was probably contained carbohydrates, and those carbohydrates have all of those carbon bonds within them, and those carbon bonds get broken down over time through a process called glycolysis, which we did not cover in this class. But your metabolism is essentially established off of the breakdown of glucose, and so we talk about glycolysis and the TCA cycle, and those are all parts of your metabolism that generate energy. And so glucose is kind of the molecule that starts the energy making process. And as that glucose molecule gets broken down, you have energy that gets released in the form of ATP. And again, not part of this course. We did not talk a ton about metabolism in that regard, but it did kind of tells you what the function of carbohydrates are. Now, lipids are a little bit different. What to know about lipids are probably the fact that they generally have like a backbone, and usually that backbone is maybe like a glycerol backbone, but you don't need to know that because that's kind of super chemistry talk. But then you've got these long hydrocarbon tails that extend off of lipids. And if anybody really does want me to pull up a PowerPoint, I can. So if me talking at you is you're not finding this helpful, and you'd like a visual, by all means. Okay. And so for those lipids, you've got your backbone, and then you've got those long hydrocarbon tails. The important thing to remember about lipids is that they can be either saturated or unsaturated. So saturated lipids have all of the hydrogen bonds saturated, and so they're long straight tails. And then unsaturated lipids have a kink in their tail because they have a double bond in there, and so that's going to cause a kind of a shape change in that particular lipid. So when you're studying about lipids, look at saturated versus unsaturated lipids, and that's again something that probably will get hit upon on the final. The function of lipids, lipids can also be broken down and used for energy purposes. Lipids compose fats, and so anytime that you remember seeing the word adipose or fat in later chapters, that's pretty much the function of lipids. So remember when we talked about bones and the fact that there's yellow marrow there. Yellow marrow is composed of lipids. Or even as we're talking about kind of how the eye sits in its socket, there's fat tissue all around your eye, kind of giving cushion. So lipids are able to kind of store energy. They're able to provide cushioning. I know people don't always think of fat as being a good thing, but the cushioning that it provides is important. And then also another function of lipid. Remember when we were talking about the myelin sheaths of neurons? That myelin has a lot of lipid in it. Those are actually fatty sheaths that cover the axons of our neurons. And so that's another function of lipids as well. So hopefully that gives you kind of, again, I know you probably are like, why are you talking so extensively about this? But I just want to make sure that I'm getting all of your questions answered. Okay, chapter 12, questions 9, 10, and 11. Okay, so question 9 is, how is a graded to potential generated? Okay, so if we talk about the difference between graded potentials and action potentials, like very generally, and this was kind of a question on the test, like a graded potential is just a slight change in resting membrane potential. So when we talk about resting membrane potential, for most cells, most neurons, it's at a negative place. It's generally sitting around -70 for most neurons. Again, that number might seem kind of arbitrary to you, but for somebody who studies it, it seems very important. But again, just remember that, you know, neurons are sitting at a negative place just because of the ionic concentration differences. And that's your resting membrane potential. And graded potentials are just slight changes, whether it be in the positive direction or in the negative direction. In the positive direction, it is a depolarization. In the negative direction, it's a hyperpolarization. And the way graded potentials are generated is literally by a presynaptic cell releasing neurotransmitter and by binding a postsynaptic cell. So if that neurotransmitter binds to that postsynaptic cell, what's going to happen is it's going to bind to a ligand-gated ion channel. That ligand-gated ion channel is going to open up, and a bunch of positive ions are going to flux into that cell, or negative, depending on what type of channel or receptor it is. And that's going to cause a slight bump up or that slight bump down. So if you have positive ions flowing into the cell, so just hypothetically, we talked a little bit about excitatory neurotransmitters. If you had a molecule of glutamate that bound to a glutamate receptor or a ligand-gated ion channel, that would be an excitatory neurotransmitter. So that ligand-gated ion channel would open. You would have positive sodium ions fluxing into the neuron, and that would cause a depolarization in that cell. So again, just a slight bump up. Now, conversely, if you had an inhibitory neurotransmitter, such as GABA or glycine, bind to its receptor or ligand-gated ion channel, that would cause that channel to open up. You would have chloride ions fluxing into that cell, and that would cause a hyperpolarization or a small, you know, dip or more negative kind of influx, I guess, in that cell. So again, you have depolarizations and hyperpolarizations, which are both types of graded potentials. If you have any follow-up questions about that, let me know. I just want to make sure that I'm being very clear about that. Okay, question 10. Describe phases and propagation of the action potential. Okay, this is kind of a big one. I don't know if I can do this without an image. Okay, so with the action potential, kind of the first step is exactly what we just talked about, which is a graded potential. So you have to have kind of enough graded potentials or a strong enough graded potential to hit what's called threshold. And so kind of the first step in that is that you have a neurotransmitter or a whole set of, you know, dendrites that get activated that cause a bunch of influx of sodium into the cell that cause a depolarization up to the point that's called threshold. Threshold, like I said, is just a point at which you have voltage-gated sodium channels open. And so once voltage-gated sodium channels open, you've got this huge uptick or rise time in your action potential, and that's what's going to kind of trigger the beginning of the action potential. So you start off with kind of a small graded potential. If you have enough graded potentials that kind of sum up, which is, again, that's called summation, sum up to threshold, then you've got that voltage-gated sodium channel that opens up, causing that fast rise time in the action potential. And at the very same time that the voltage-gated sodium channel is opening, you also have voltage-gated potassium channels that open. Now, if we remember kind of about which way ions are moving based off of their concentrations, sodium is always going to want to go into the cell because it's got a high concentration outside of the cell. Remember, salty banana. And then potassium is always going to want to, you know, go out of the cell, which is going to cause the cell to become more negative. So that voltage-gated sodium channel causes the channel to, or the voltage change, the threshold reach, causes the voltage-gated sodium channel to open. You get that high rise time. And then as you get to that peak there, the voltage-gated sodium channel becomes inactivated, and then your voltage-gated potassium channel opens, and that's what causes your downward slope of the action potential. And because that voltage-gated potassium channel is so slow to open and so slow to close, there's this kind of dip that goes even below resting membrane potential, which is called the after hyperpolarization. And that's kind of right below that resting membrane potential. Now that's kind of important because it's below resting membrane potential, the cell's actually sitting a little bit more negative than it otherwise would be, which makes it very difficult for it to fire another action potential at that time. Okay, hopefully that answered that question. And again, if you need any follow-up questions or if you need me to repeat something or slow down, make sure that okay. Question 11, describe signal transmission at the chemical synapses. Okay, so signal transmission at the chemical synapses, just very quickly and briefly, is that you have an exponential traveling down an axon, and it gets to the very end, which we'll just call like, this is the presynaptic side of the cell. So you have a presynaptic neuron that is going to be, I don't know if I want to go into this much detail, but I will. You can kind of close your ears if you like. But once you get the action potential that travels down to the presynaptic side, you have voltage gated calcium channels that open. Okay, we've talked a lot about voltage gated this, voltage gated that. Voltage gated calcium channels open, causing calcium to influx to that presynaptic cell. And the influx of calcium is what stimulates the release of neurotransmitter from that presynaptic cell. You have neurotransmitter being released, it diffuses across the synapse, and then it binds to the postsynaptic cell, which has receptors or ligand-gated ion channels, as we've learned about them. That's what they really are, ligand-gated ion channels, or maybe just ligand-gated receptors. Receptors probably the best because I won't get into why that is, but you have receptors on that postsynaptic cell that are going to receive that neurotransmitter message. Now, in terms of kind of terminating that message, there are ways in which that happens. You can either have enzymes in the synapse that kind of break down the neurotransmitter and cause it to, become components that won't stimulate cells, or you have the astrocytes that we learned about that have little kind of like neurotransmitter vacuums on them. And so when neurotransmitter gets released into the synapse, those vacuums kind of get turned on and they suck up all the excess neurotransmitter. So you just don't have neurotransmitter hanging out in the synapse for a super long time, kind of, stimulating cells. Kind of an interesting, like we're talking about transporters sucking up that excess neurotransmitter. If you've ever heard of SSRIs, which are selective serotonin reuptake inhibitors, that's a particular type of drug that blocks serotonin reuptake or serotonin transport, which causes serotonin to hang out in the synapses far longer, and then thus kind of stimulate serotonin receptors much longer. And so that's why it works effectively as a mood enhancer. And actually cocaine does the same thing. It blocks dopamine transporters, and then you have just a ton of dopamine in your brain kind of exciting those receptors, which it makes sense why that's highly addictive. But again, that's outside the purview of this course. Okay. Again, if you have follow-up questions, please make sure that you're asking those questions. What causes the after hyper? Okay, I'm gonna keep going down the list. I'll get back to that, Addie. Well, maybe not, because we just talked about that. So the after hyperpolarization, what causes that is the fact that you have those voltage gated potassium channels that open so slowly that they only cause that downward trend in the action potential after the voltage gated sodium channels have opened. But those potassium channels are very slow to close. And because they're slow to close, you basically have a ton of potassium leaking out of the cell. And so that all that potassium leaking is causing the cell to become more negative. So it becomes slightly more negative than it would otherwise be in your kind of normal general resting membrane potential state, if that makes sense. Okay, questions 7 and 8. So Addie, let me know if that answered your question or if you need me to follow up more. 7 and 8. Okay, so describe the clinical significance of dermatomes. So dermatomes, again, are regions of the body that map directly to, let's say, the dermatomes are regions of the body that directly map to specific, what's the word I want to use here? Specific regions that innervate your spinal cord, even the specific plexes. And so you've got a specific region, let's say on your hand, that maybe maps to a nerve that projects up to a specific, plexes, or a specific ganglion is probably the most proper word to use here, which is just a cluster of cell bodies that sends the information up into your brain. So dermatomes are important because if there is an area of damage at a specific dermatome, it allows you to map out exactly where maybe the nerve damage has occurred, let's say in the spinal cord. So if you have, let's say, a region of the body that is being non-responsive to some type of stimuli, so let's say if you have a part of your leg or a part of your arm that it's no longer transmitting, let's say, pressure information, temperature information, or what have you, to the spinal cord, and you're not receiving that message in the brain, it helps you kind of start to determine where in the spinal cord or where in that nerve pathway there might have been some type of damage. And so because our bodies are kind of mapped so kind kind of clearly that way, you can actually kind of map down pretty specifically which nerve or which nerve ending. So let's say if you have a compressed nerve or a pinched nerve in your spinal cord and that's correlated to a specific dermatome, you can actually diagnose that in that manner, if that makes sense. 7 and 8. Describe the distribution of cervical plexus. That's okay. The distribution of the cervical plexus, brachial plexus, lumbar plexus, sacral, and coccygeal plexus. I feel like we almost should just go chapter, is that chapter 14? No, that's chapter 13. Seeing if I have that pulled up here. Yeah. Ha, ta-da, found it, okay. Plexes, okay. And I will say this, like generally I appreciate the folks who attend these study sessions. So with that, generally the conversations that I haven't written the exam yet, because I sometimes even just like to come back to this review session and see kind of what I've spent my time talking to you about. It's just so this becomes even more worth your time attending, if that makes sense. So when I'm writing those exam questions, I'll probably go back and rewatch this video, or even go back through our chat and see if, okay, well, we talked a lot about this, we talked a lot about that. So those folks who attended, you know, I want to make sure that, again, that we're making this worth your time. Okay. Okay, I'm going to keep working my way through kind of these questions. So question 7 and 8. So cervical plexus, again, we're talking about kind of the neck and shoulder region. Again, this isn't like a super interesting question because I think it's very much just, I'm not going to ask you about like C1, C2 versus C3, C5. I'm going to keep it very generalized to these regions. So the cervical plexus would be, you know, skin and muscles of the head and neck, and the shoulders. Your cervical is going to be your upper kind of shoulders as well. Oh, sorry. So your cervical plexus. So you can see here some of the branches of that and kind of where they end up: neck, shoulder, things like that. Infrahyoid muscles of the neck, things like diaphragm. So that's also going to be kind of important. I think maybe that was a question on the last exam, asking if you could determine where there was an area of damage. And I think one of the descriptions for that particular patient was the fact that they were having difficult breathing. And again, because they were having difficult breathing, that tells you that the nerves that were stimulating the phrenic nerve that sent information to the diaphragm could possibly be affected. The brachial plexus, again, you got upper limbs, and you can kind of see that an image of that here. Your lumbar plexus is just going to be kind of those lower limb, you know, lower trunk limb types of regions. Again, I'm not going to get into super specific regions here just because, one, I don't feel like we spend a ton of time here. And so I just kind of want you to walk away with kind of a general understanding of this. And lastly, the sacral plexus, which has, you know, again, regions of the lower limb that it's innervating. But again, I'm not going to go into like a ton of specifics. So I would say don't pull your hair out too much over the plexus, as long as you can kind of look at the laundry list of possible muscle regions that are innervated here and feel comfortable with those, and you should be fine. But again, I wouldn't go too crazy spending a ton of time, especially on chapter 13, just because we didn't spend a ton of time there during the semester. So I don't necessarily know if it's a great use of your time now. Okay. Okay, I'm just still going through the laundry list here. Okay, the effects of GABA as a neurotransmitter. So GABA is an inhibitory neurotransmitter, which means that it causes the cell to become more negative. And so GABA will bind to a ligand-gated ionotropic receptor, or sorry, shouldn't use that word, a ligand-gated channel. And once that neurotransmitter binds to that channel, it causes it to open. There's an influx of chloride into the cell, and it causes a hyperpolarization or a more negative kind of change in the cell's resting membrane potential. So you'll see a little dip. And because of that, it makes it more difficult for an action potential to fire. So the reason it's inhibitory is because it's preventing an action potential, because you're lowering kind of the internal resting membrane potential of the cell for just a split moment. So it's harder for it to fire an action potential, even if it has positive ions fluxing in the cell somewhere else. Okay. Okay, so chapter 4, questions 1, 3, and 7. Just need to get back to our study guide here. If it's even on my page. Okay, so chapter 4, question 1, 3, and 7. Okay. Name the 4 basic types of tissue that make up the human body and state the characteristics of each. Okay, so the types of tissues that make up the human body. Okay, so again, this is a pretty general question. So you have nervous tissue, which we've talked about extensively. We have epithelial tissue, which we, I guess, connect. Okay, so I think we talked about connective tissue, epithelial tissue, nervous system tissue, and then muscular tissue. And I think for chapter 4, we talked far more about epithelial and connective tissues than the other two, because we had chapters kind of associated with those other two after that. And so the characteristics of each. So if we're talking about epithelial tissue, like there's a lot of different kind of shapes and cell types that are associated with epithelial tissue. I would say epithelial tissue most often creates kind of those that we learned about junctions in that particular chapter a ton as well. So epithelial tissue generally like lines things and functions primarily in absorption or secretion. So I think when I think of epithelial tissue, kind of the characteristic type of tissue that I think of is the tissue that lines our digestive system. But we just learned about the corduroy plexus in the ventricles, which is also a type of epithelial tissue, because it lines those large, regions that are open spaces. And again, the purpose of that corduroy plexus is to secrete cerebrospinal fluid. So that's a function that kind of falls under the epithelial tissue kind of characterization. Connective tissue, as we saw, is going to function primarily, again, in connecting different regions of the body. So we learned about things like basement membrane. Connective tissue is located, in the lining of different organs and things like that. But we talked, again, kind of about the overall structure of connective tissue in chapter 4. Nervous cell tissue, we talked about extensively when we talked about chapter 12 and the different types of nerve cell types. So we talked about neurons and astrocytes and kind of how they're grouped out. And then lastly, muscular tissue, we kind of skipped over chapter 10, which would have gone into, I think, a little bit more detail about how muscle tissue is essentially characterized, whether it's smooth muscle, or muscle that's used for contractions and kind of is more present on like our in regions that we use for movement. So again, things that we haven't necessarily talked about extensively, you can consider to be pretty much free and clear of on that final exam. The main differences and general features between epithelial and connective tissues. So probably some of the big differences, I think, like if you were going to like look at epithelial tissues and connective tissues, epithelial tissues are kind of just like little, I think of them as little boxes, but they can really have a lot of different shapes, but they're generally kind of stuck together very, very close, and packed together very, very close. And if you look at connective tissue, those cells don't look like that at all. They look very large. There's like these different kind of structural components that are kind of helping them keep like this large kind of almost like tent-like, they look like tent poles within the cell. So that's going to be kind of one of the main kind of structural, very featural differences. Again, epithelial tissue has a plays a much greater role in secretion and absorption, and connective tissues kind of connect different areas. So they have both structural differences, and because of the structural differences have different functional differences. But again, that's kind of the level of understanding for epithelial and connective tissues that you'd need to know. And it's probably worth just going back and spending a little time on chapter 4 if that was not necessarily your strongest chapter. Because there are kind of, you know, sub structures within connective tissue that we can talk about more. That's actually the next question. So describe the structure, location, and functions of each of the different connective tissues. Okay, this we might have to go back to, go back to the source, which would be getting the actual PowerPoint pulled up here. So sorry, it's going to take me a pause here for a moment to get that pulled up, just because I don't want to forget one hypothetically. But I guess as I'm getting that pulled up, we'll see. Is there, how much will we need to know about specific disorders for the specific chapters for the final? Let's see, unless I spent like a ton of time talking about a disorder, which I don't really think we did a ton for any specific disorder. I would say don't necessarily spend a ton of time there. Like most often I won't, if I start to describe a disorder, I'm going to give you kind of a hint about something else within that disorder that relates back to the information. So if we're talking about a disorder that it's a dysfunction of the specific tissue type, I'll probably talk about that specific tissue type. So even if you don't know the disorder super well, you should hopefully be able to deduce the answer given what you know about that particular tissue type. Yes. Okay, and I'm going to answer this other question before I go back to finish is there anything that's going on to be on the test that you suggest we study more than other things? Like I said, going back and looking at the previous exams is someplace I would definitely spend my time. And like I said before, I'm going to try to probably pull from different chapters together to kind of show how the chapters integrate to one another. I mean, I think probably the farther back the information kind of goes, like chapter 1, 2, and 3, because that is so far back, I'm probably going to ask you far more general questions about those chapters that are farther back, and then probably be maybe a little bit more specific in the chapters that we've more recently gone over. I guess that's kind of, you know, like right now, like I just kind of alluded to, is like the material that we're going over in this study session, I'm going to try to go back and intentionally ask questions about the stuff that we're going over now. So I'm recording this, so it probably is worth your time, if you're not taking notes now, to go back and watch this video at that later time. And essentially that's what I'm going to do, is go back and actually write probably a good, as many as I can, questions about the material that we're kind of talking about right now. So start asking me questions about super easy things, and then that's what we on the final. No, don't do that. You'll be like, what's that bone again? Okay. Just want to make sure I can get this. Okay. So the question that I'm answering now is basically about the different connective tissue cell types. And so I just wanted to bring up this particular slide, just because I think going back to this chapter, looking at this particular slide, I think like this is probably the slide to take away for the connective tissue cell types, just because it shows you how they're all interacting with one another and kind of their functions as well. So recognize when we're talking about connective tissue cell types, and I think I did the circling kind of and grouping before, and maybe you'll find this helpful too, is like there are definitely cell types on here that are immune cell types. So if we talk about things like macrophages, fibroblasts, mast cells, plasma cells to a certain extent, neutrophils, eosinophils, are all different types of immune cells. And so their function is going to be to mediate an immune response. And so often it's finding the foreign invaders that are in the body and going out and fighting them, or sometimes it's facilitating an inflammation response. That's kind of, again, the main function of the immune system. We can see other things that are more structural in the types of cells that they are. So things like reticular fibers and collagen fibers and elastic fibers and ground substance. Those are all far more kind of structural cell types. And so if I was going to go back and kind of focus on learning connective tissue cells, I would literally put these into like 3 different columns and just make sure, because I think if you kind of know the general function of the cells, it's kind of easier to also remember what they do. Adipocytes are just going to be your fat cells. Again, plays a role in structure and cushioning to a certain extent. And what other cells have we not talked about? I think we hit all the cells here. So again, you've got cells that function in structure, cells that are functioning in immune response. And those are kind of like the 2 big, I think, categories for the connective tissue cell types. Were there certain questions on exam 3 that most people missed? There were a couple of questions, I think, that were, I think, more difficult on exam 3 than I think people were kind of bargaining for sure. But I don't, let's see, I can keep asking me questions and I can actually tell you which questions folks had issues with right now. As we're chatting. Will there be a lot of labeling like in exam 2? So let's see, I said 40 questions will be from previous exams, and we've had essentially 3 exams up to this point. So a third of 40, whatever that is, you know, roughly like 10 or so questions, we'll say 10 to 12-ish questions, is going to end up being labeling like that. So yes, there will be a certain chunk of questions, but recognize that those questions are taken from those previous exams. So that should hopefully kind of narrow down which kind of labeling, which labels to kind of study, I guess, if that makes sense. Okay. Keep asking questions. Okay. Because I'm still looking through which questions did people have issues with. Let's see, that one looks a little low. I really can still answer questions as I'm going through this, so. Okay, somebody asked about white and gray matter. Okay, so the easiest way to, I think, to memorize the difference between white and gray matter is to think of white matter as fat. Okay, white matter is going to be fat. If you think about white matter as fat, think about where the fat is located on the neuron. And the axons of the neuron are myelinated with fat. I mean, if that helps you kind of, again, think of it in a very, very simplistic way. So axonal fibers are always going to be white matter because they're covered in fat, and gray matter is always going to be cell bodies. And so the function of the spinal cord of gray matter. So the gray matter in the spinal cord contains cell bodies that are going to kind of mediate the responses of the spinal cord. So most of the, I'm not going to say most, but some of the gray matter, some of the cell bodies that are located within the spinal cord are called interneurons. And so they're essentially, those interneurons are kind of acting, we'll say like relay stations, and they're deciding whether they're going to send information back out into the muscle or whether they're going to send information up to the brain. And so they kind of get, I guess relay station might be the best way to put it, that they are they're relaying the information either back out into the muscle or back up into the brain. And so those cell bodies there are mediating that response, if that makes sense. I don't know if there's any better way to kind of describe it, if that makes sense. So the function of the cell body in the gray matter. So I recognize that some people are direct messaging me. So people might be like, which question was asked? The question was, what's the function in the spinal cord of gray matter? And so the function of gray matter exists because there's cell bodies there. The cell bodies in the spinal cord are relaying messages either back to the muscle or sending messages kind of up to the brain. And recognize the cell bodies are located in the gray matter and they're projecting out into the white matter, and the white matter is going up and down to and from the brain. Okay. Okay, this question, I guess, On exam 3, you are clicking on your computer right now. As you touch your mouse or mouse pad, what type of receptors are being activated within the skin of your fingers? This one people kind of had an issue with. So the receptors that are located, the pressure receptors that are located in the pads of your fingers are mechanically gated channels. And so as you're pressing on something that's mechanically gated, they're causing kind of a physical distortion of those channels, causing those channels to open up and essentially cause a depolarization and send an action potential or a graded potential to your spinal column. So that was one question that folks had issues with. And then the next question was—
They send graded potentials, not action potentials? Or?
It depends on the strength of the stimulus. I guess is ultimately kind of the answer there. But if the spinal cord is receiving the message, most likely an action potential is being fired there in order to kind of sustain that information being sent all the way to the spinal cord. Okay. The region of the cerebrum that generates output for the somatic nervous system. So that would be, that was the other question I think that folks kind of had an issue or like didn't necessarily have the easiest time with. And so that's that generates output, I think was the important question here for your somatic nervous system. So your somatosensory cortex is going to be receiving all of the input from your peripheral nervous system. Somatosensory cortex is going to be processing it. And then in terms of output or that's being sent to your somatic nervous system, that's going to be coming from your motor cortex. Your motor cortex is going to mediate the types of motor response. And so when you hear the word somatic nervous system, you should think musculature or muscles. And so your motor cortex sends information out into your somatic nervous system. I'm guessing a lot of folks chose B on that one just because they saw the somatosensory kind of in similarities in terms of what's the word? Just nomenclature. Okay, someone asked what the average on the test was. I'm getting there. Okay, and again, please feel free to continue. So the average for this exam was a 75, which is a little lower than some of the previous exams. Okay. Other questions or concerns? Okay. Do you think it comes through of interstitial, extracellular, cytoplasm, intracellular, cytosol? Okay, so I think that nomenclature could be confusing because it's used in a lot of different senses. And I think it almost depends on what type of person or scientist or whomever you're talking about that will use those different kind of terms, maybe interchangeably. So interstitial fluid is going to be more the relationship of the fluid between an organ and an organ system. And when you're talking about things like connective tissue, so interstitial fluid would be kind of the fluid that kind of bathes the outsides of organs. Extracellular solution. So if we're talking, I think the most often when you hear the terms extracellular and intracellular, those would probably be used more frequently when you're talking about cells, or you're looking at like an individual kind of cell type. I think I probably use the term extracellular and intracellular a lot because that's how we refer to things in the nervous system kind of world. You think about intracellular being inside a nervous cell, extracellular being the outside of the cell. And because ionic concentrations for those two things are so important, that's how we think, that's kind of how we give those names. Cytoplasm is going to be basically, when we talk about cytoplasm and cytosol, that kind of harkens back to chapter 1. The The cytosol is going to be kind of the liquid gelatinous material that's inside of the cell, and the cytoplasm is going to be kind of everything encompassing that, including the cytosol. So I think the cytosol plus organelles equals cytoplasm. So again, it's just kind of a very delicate nomenclature dance. But I mean, if you need the answer to that question just for like references as you're going through your material, then I hope that kind of gives you that. But for the most part, I don't think I'll be necessarily asking you to differentiate those things. So maybe that's okay. Over bone cells. Okay. Bone cells, we have osteoclasts, osteoblasts, osteocytes. There's another one, osteoprogenitors. I think that sounds right. Yes, osteoblasts. Yeah, okay. So your osteoprogenitor cell essentially can differentiates to become kind of the more mature bone cell type. Blasts are going to be cells that are kind of in the active process of secreting, matrix in order to create the structure of the bone. So osteoblasts are the building cells. They're secreting this matrix. They're allowing the bones to be made. And then osteocytes, if you see an osteocyte, it kind of looks like a dehydrated, almost to a certain extent, osteoblast, but it provides kind of the structure for the cell. So that's the osteocyte is going to be the mature bone cell. The osteoblast is the building bone cell. And then lastly, the osteoclast is going to be the bone cell that is actually derived from a whole different like lineage. It doesn't come from where the other bone cells come from. It actually comes from some immune cells. And the reason for that is because it has some properties that are similar to different immune cells, where its function is to break the bone down. So it's secreting enzymes to break the bone down. And if you remember kind of when bone is being built and remodeled, it's all about that balance between building and breaking down. So osteoblasts build up, osteoclasts break down, and that's even kind of how the bone, the follow-up question is how the bone grows in length and thickness. And the way that bone grows in thickness, kind of the girth of the long bone at least, and that's kind of the example that we went over, is that you have osteoclasts that are eating the center of the bone out. And then you have the osteoblasts that are kind of building on the outside. So it's almost like if you had a tree trunk, if you were kind of gnawing your way through the center of the tree trunk on your all the way out, and then you're just kind of throwing on additional layers of tree on the outside. So I know it's the metaphor is probably not a perfect one and not that helpful, but just to kind of think of it in a different respect. So again, osteoblasts. And then in terms of kind of how the bone grows in length, you've got your epiphyseal plate and your epiphyseal line, that is your growth plate in your long bones, and you just kind of have the additional addition of bone cells that occur there, beginning as connective to as kind of immature bone cells, and then maturing over time and kind of again expanding out the length of that bone, if that makes sense. I guess I could pull up an image from that chapter, but you have about 6 minutes left if anybody has any questions. Okay, the question is, can we go over resting membrane potential? Sure. Okay, so resting membrane potential is essentially a product of what we like to call the electrochemical gradient. And so the electrochemical gradient refers to both the differences in electrical charge between the inside and the outside of the cell, and then also the differences in concentration. So the cell kind of constantly is functioning to maintain this negative resting membrane potential. And so the way that it does that is by kind of two different things. So first is the sodium potassium, either ATPase or exchanger. There's a lot of different names for it. The sodium potassium I guess ATPase is good because it kind of describes exactly what it does. And also the potassium leak channels. Kind of those two things are going to be the things that can contribute to resting membrane potential. So you've got a high concentration of potassium inside the cell, and it's constantly trying to, again, because you have a high concentration, it's trying to go down its concentration gradient from an area of high concentration to a low concentration. So because of that, every time those leak channels, those potassium leak channels open, you've got potassium leaking out of the cell, making it more negative. Now, as that is also occurring, you have the sodium potassium ATPase, which is constantly pumping sodium out of the cell and pumping potassium into the cell. So that particular exchanger is part of just consumes a ton of ATP. So when I say ATPase, that's why, because every time that exchange happens, you utilize ATP. So every time it's pumping 3 sodium out and 2 potassium in, it's causing that difference in the difference in the concentrations, but then it's also utilizing a ton of energy. So it's generating that difference in sodium and potassium concentration gradients, but then it's also kind of contributing to the effect of the potassium leak channel. So every time that potassium leak channel opens, potassium's leaving. So that combination, again, I'm giving you those numbers of 3 sodium leaving and 2 potassium coming in, because every time that exchange happens, you essentially are kind of netting one negative charge. And so kind of all of that net negative charge adds up to that minus 70 millivolts that we get. And again, that minus 70 millivolts is, you probably don't really care and won't really ever memorize it, but again, it's important to someone like me, and it's just a kind of that net negative charge of where the cell is sitting. Why and when did the leak channels open? The leak channels, I mean, it's like having like a leaky faucet. Like when and why does a leaky faucet leak? It doesn't really have a rhyme or reason to it, right? Because for most faucets, you think of them having as like on and off switches, but if you have a leaky faucet, just happens when it happens. And so that the answer to those leak channels is it's sporadic, spontaneous, and at any time. Like those leak channels, since they're not necessarily gated by anything, they'll just randomly open and close. And so there's really no rhyme or reason to those leak channels. But what's going to affect how they work, or the effect of those leak channels, is going to be that concentration gradient. So if you have a high concentration on one side and you have a leak channel that opens, there's going to be a larger, what they call driving force, or a lot more force causing, more ions to leave. It's like if you have a ton of people in a crowded room, every time you just randomly open the door, if you have a very crowded room, you have a probability of a lot of people kind of shooting out at that random door open. But if there's like maybe like 5 or 10 people in a pretty large room and you're randomly opening and closing that door, not very many people are going to just randomly kind of wander out of that room. So just whenever you're kind of thinking of ions, you can always again think of your if you're maybe an introvert, antisocial person, which there's nothing wrong with that, but I'm saying think the ions are very much the same way. They're always trying to get away from their bros and get to an area where there's not as many folks. And so that kind of is the same thing for leak channels too, right? Random opening and closing. If there's a lot in the inside, you've got a lot more what they call driving force, forcing ions out. I know I always use the club analogies, and I'm like, there's not even a club in Montana, really. Yeah, but I feel bad going, like, if you're ever in the bar, I feel like club is just, it feels like a better to say that than the bar, but same probably difference, really. Okay. Thomas, the question that you asked was, when will you get your exam grades? As soon as we wrap up here, I'm going to release your exam 3 grades too, and then hopefully start working on opening up the lowest score for everybody to make those corrections. I think we've lost a few folks, but that's okay. This is kind of your last chance in terms of asking questions, concerns, what have you. Yeah, well, hopefully that can happen today. But I don't know, since you weren't here at the beginning, I'll be perfectly honest, like I've been trying to import your grades into Moodle, and every time I do that, I overload the system and it locks me out of using that type of browser. So I basically have locked myself out of Chrome and Firefox, and now I'm working through Safari, and I didn't do it in Safari so I could actually access our files while we were doing this. when do we have to have the corrections back? I'm going to probably push the corrections. I'm going to see when grades are due. I would hypothetically like the corrections to be due by the time of the final, but kind of depending on when the corrections kind of get opened up to you, that might change the due date for that. So I want to make sure you get plenty of time to go through that. But I also think that it would be very helpful to do that prior to the final, using it kind of as a study technique. So it kind of makes more sense for you to do it before the final. Yeah, I'll just kind of see when grades are due or when the end of finals are. Does anybody have any idea what the last day of finals are? Because we could just probably make it that day, honestly. 24th, the next day. Okay, so probably the 24th, 24th, 25th. Okay, I'm gonna just go a quick Google right now and we'll come up with that answer. Okay. Any other questions? Finals week schedule. So here we go. Are there finals on weekends here? Oh, okay. Yeah, thank you all so much. I've had a really fun, okay, it looks like the 24th is the last, Wednesday the 25th, I see some schedule though. Yeah, so Wednesday, probably the 25th. Thank you all so much for anyone who's still around. I'm sure you're probably all trying to head out. Thank you all so much. I've really had a fun time, even though this is kind of a weird setup. This has by far been one of my most fun classes in a weird way. So thank you all so much for the time and effort that you've put into this course. I really appreciate it. And if you have any questions or concerns between now and the final, do not hesitate to reach out to me via email. I'll get back to you as soon as I can. Make sure you're taking a good hard look at your grades in Moodle. And if you have any questions about that or you need any types of auditing done there, we did end up kind of coming up a little short in terms of the number of adaptive practices, I think, that we got assigned for this semester. So we might be, there might be one or two of those that kind of get cut off. So the total number of points for the course might be a little lower than 1,000. So just take that into account. And I haven't put in any of the extra credit assignments that have been turned in yet. So don't necessarily just recognize that those aren't really accounted for yet. So just again, remain patient. I am focused on this course and getting all of the grades in as soon as possible, because I know that it's very important for you to know this as soon as possible. Does the count? So for chapter 10, those adaptive practices and quizzes haven't been put into Moodle yet. But that will be 10 for your quiz, 10 for your adaptive practice. And then if you turned in the writing assignment, that will be another 20. So there's 40 possible extra credit points out there right now. I guess everything should be turned in by now though. Okay, does anybody have any other questions or concerns? Yeah, no problem. Okay, well, that's all I have for you. If you have a specific question that you'd like to talk to me about one-on-one, you can definitely hang out. I'm going to stop the recording now, and yeah, everyone, good luck with finals. I hope you do amazing.